So, thank you very much. I apologize because I speak English. My French is not so poor, but I couldn't give a presentation in French. So, thank you for inviting me, and I only have about 15 minutes to talk about the exposome, which is a very broad concept. So, I will try to describe some of the research I have been involved in, with some emphasis on the social exposome, because lately, in the last few years, I led a network about let's say, the social exposome, and what we call the social to biological transitions. So I think it's not necessary, of course, to remind you the definition of the exposome. What I just want to stress is that you find now definitions also in dictionaries, so it has become a common word, so to say. And the different definitions, well, they tend to be a little heterogeneous, but essentially they say the same concept. That is, it is something that has to deal with the life course. So it is about the influence of your life course exposures since conception, in fact. And also it includes external exposures and internal changes to influence the outcomes along the life. As you certainly know, the exposome was probably initially conceived as having to deal with chemical and physical exposures, but very soon Chris Wilde and others included the social components, which can be interpreted in several ways, like social inequalities, for example, social networks, stressful experiences. Now we are talking much about the childhood adverse experiences. So stressful experiences starting early in life. This is a way to represent the exposome going from the external exposures that include, well, in practice everything, diet, water contamination, air contamination, social relationships and stressful conditions. Then you have what is called the internal exposome, which is based very much on the new developments of omic technologies, for example, or epigenetics. I will show something, for example, about the epigenetic clocks, which is one of the developments. And finally, you have the outcomes, but also the field of the outcomes has become more complicated because the classification of diseases is changing. There are new ways to look at multimorbidity. Think of the idea of syndemic, which has been applied by Richard Horton to COVID. And in general, the idea is that it is a little artificial to isolate single disease entities. And non-communicable diseases have common roots, common mechanisms, and the direction now is to go to look at multimorbidity. So just a few examples. This is about the diet. Some of the examples I show refer to the improvement of exposure assessment, which is one of the goals of the exposome. This is research done at IARC based on the EPIC cohort, a very large cohort in Europe that was recruited in the early 90s. This is mass spectrometry in urine in relation to dietary habits. And it is just to show that with this approach, you can clearly distinguish different types of dietary items like coffee, wine, orange, chocolate. You see that the spectra, the profiles are quite different for each type of intake, of food intake. So this is a way, as I said, to improve exposure assessment in a field which has always been extremely difficult, diet. Questionnaires on diet have many limitations. In principle, you have to cover decades of dietary intakes to investigate in a meaningful way the impact on health. So there is a convergence between epidemiological research and laboratory research in this way. This is very new, very, very new. This is research done at Imperial College within the section I lead, which is called Molecular Signatures and Pathways to Disease, and in the MRC Center. And this is research done by Leon Barron on the water exposome. What they do is to measure, once again with mass spectrometry, chemicals in rivers around London, and to characterize the exposome, the external exposome of such rivers. And they are extremely interesting findings. Obviously, they find a lot of illicit drugs. They started from that. But then they find pesticides, industrial chemicals, and each river tends to have its own profile depending on the exposures, on the upstream sources of exposure, like industries, agricultural activities, even home use of pesticides and drugs, many illicit or not illicit drugs. So the next step will be an attempt to link this water exposome with disease and internal measurements in people. This is still another way to look at the exposome. This is mutational signatures. In this case associated with tobacco smoking. Just to say that you can also look at mutations. People usually think of the exposome in terms of, say, biochemistry or metabolomics, metabolites. Well, in this way, we have characterized the mutational signatures in biopsies of people with different types of cancer, and we found a mutational spectrum in lung cancer, which was the same found in cells treated with benzopyrene, which suggests that benzopyrene might be or PAHs would be the responsible agents for the induction of lung cancer in smokers. Well, this is another concept which is inbuilt in the idea of exposome, that is by investigating certain profiles in people with disease and profiles in people exposed to certain, in the same people, in relation to exposure to chemical agents, you can find an overlap. In other words, this is on the right an example of metabolomics in two cohorts, Sapaldia and EPIC, in relation to air pollution. These are longitudinal studies. So what we did was to characterize exposure to air pollution through metabolomics in blood collected about 20 years before the onset of disease. And then we looked at asthma and cardiovascular diseases, and we found an overlap for certain molecules, indicating, obviously this is not surprising, inflammatory pathways. It's not surprising, but it is, let's say, proof of concept of the idea of meeting the middle. That is, you can find overlapping changes which lend support biologically to the causal interpretation of relationships between exposures and diseases. And the social exposome, as I said, I have led a Horizon 2020 project called Lifebutter, which was in a way unusual because we had a very strong social sciences component, particularly in Toulouse, Cyril Delpierre, Michel Kelly, Irvin, Thierry Lang. So I learned a lot about social sciences. I became very fond of Pierre Bourdieu. When we developed some new maybe they are not so new, but some concepts of interaction between the biological approaches and the sociological approaches. So our thinking that is expressed, for example, in a paper I wrote with Michelle Kelly-Irvin, is that in Pierre Bourdieu's concept, which is very well known about the social capital, cultural capital, and economic capital, what is missing is the biological capital. There is something, if you read the very difficult pages, I must say, on the concept of habitus, but still a formalization of this idea of biological capital in relation to the other three types of capitals would be extremely interesting. And our proposal is to connect biology, zoe, and biography, bios in Greek, via the social exposome. So the social exposome is this kind of social to biological transition that can be investigated through social sciences, but also with all the other tools offered by the exposome. And well, what irritates me very much, for example, is that in the biomedical sciences, sometimes the social sciences are looked at as something not interesting, or soft science instead of hard science, and we need to overcome this attitude. So just to give a couple of examples coming from Lifebutter, one of the ideas we pursued, and in addition to the use of metabolomics and epigenetics, was also the concept of allostatic load. And this is, in fact, research done by the Toulouse group on the measurement of different biomarkers that contribute to the creation of an allostatic load overall marker. And we found a very strong association between the allostatic load measurement and mortality. Well, allostatic load is essentially the idea of resilience of the body or different organs to stressors. So there is a degree of resilience based on metabolic and molecular responses, but this ability to cope with stressors at a certain point is overcome, either because stressors are very frequent or because there is a long-term exposure. And obviously, individual susceptibility is key there. So we showed that allostatic load was associated with socioeconomic status and also with mortality. And then we used the idea of biological clocks based on methylation, on epigenetic changes. So there, you may be familiar with this or not. In any case, the original concept was developed by Horvath in California, who found that if you look at changes in 353 CpGs, hypomethylation or hypermethylation, usually hypomethylation, these changes are strongly associated with age, chronological age, with a very high correlation coefficient. But still, there is a discrepancy between the clock, the methylation clock, and chronological age at the individual level, when you go from the population to the individual. And this is called age acceleration. So everyone has a degree of discrepancy between methylation of CpGs and chronological age, which is an expression of age acceleration or deceleration. So it's not difficult to imagine the implications of this idea of age acceleration. And one of the first things we looked at was whether age acceleration was associated with socioeconomic status, and it was. This was done in 3 cohorts. MCCS is Australian and Tilda is Irish. And if you just look on the top right, the baseline is a high socioeconomic status, and you have age acceleration in the intermediate category of socioeconomic status, and then higher age acceleration in the low socioeconomic status category. After adjusting for behavioral risk factors for chronic diseases like smoking, obesity, hypertension, and diabetes, and so on. So we expanded this kind of research with a joint analysis of 18 cohorts in the world, cohorts with methylation and information about risk factors. Once again, we looked at age acceleration after adjusting for behavioral risk factors for non-communicable diseases. Well, just look at the blue points. There you see 4 different types of clocks. Just look at the blue one, which is one of the most recent. Now there is a kind of proliferation of epigenetic clocks, including a metabolomic clock that we are working on. So you see that there is an association between socioeconomic position and age acceleration, once again, irrespective of other behavioral risk factors. Obviously, smoking, BMI, low physical activity, they have an association with age acceleration. But there is also an additional association related to socioeconomic status, which goes together with the idea of Michael Marmot, for example, and others, that there is an impact of social position which goes beyond behavioral risk factors. And Michael Marmot was also a partner in this network. Final slide. What are the gaps? Clearly, there are many opportunities in the idea of Exposome. And personally, I like very much the idea of an expansion to social determinants of health, and this idea of social to biological transition, which has also been called embodiment by Nancy Krieger. But there are several gaps, conceptual gaps. For example, if you take the hallmarks of disease, there are the very well-known hallmarks of cancer by Anahan and Weinberg. Well, the temporal sequence is not clear at all, and through the exposome you can clarify it. It's not only mutations and epigenetic changes, but also the role of immunity, cell proliferation, and so on. Then what is the environment? And now we have a more complex and maybe sophisticated idea of the environment. It's not just the biomedically relevant events that we are used to, like infections or chemicals or physical exposures, but also the social exposures. Then the life course approach has only been partially explored because we don't have, in fact, cohorts that go from conception on, or multi-generational cohorts with very few exceptions. So one is E3N and E4N here in France. Then diagnostic approaches are still immature, for example, because there is an incomplete coverage with the usual platforms of CPGs in the case of epigenetics, or annotation of metabolomics is very, very limited, and so on. And there are rapid developments of in molecular technologies. Some of them are applied to epidemiology. For example, non-coding RNAs. Now there is much research on those, but often costs are very high. There are still technical issues like preservation of samples, degradation, and so on. Then there is an interesting development also in sensors, but we have not gone very far, I would say. And by sensors, I mean portable devices to measure external exposure. And there has been a lot of development in biostatistics, here in France with Rémy, for example, at Imperial with Marc Chadeau, who is French. But there are still gaps in crossomics in the relationship between different omics, mainly due to the very large numbers of signals, which are much higher than the numbers of subjects. So there are obviously very concrete methodological problems to solve. Thank you.